this is the tree that we choose for our practical one. We call it Pokok Pagar Anak. So this is the sample that we have taken from the tree. We have 10 leaves in the sample, but we make it like 3 sets. So we have calculate the width and the length of the leaf, and next we will show the data that we have right.
Now we are going to take a look at the result for practical 1, which is basic concepts of statistics and variation. All the data for set 1, 2 and 3 were collected and used for calculating the mean, variance and also standard deviation. The formula for the mean is sum of the data divided by the number of the data. The variance is calculated by taking average of squared deviation from the mean, while the standard deviation is square root of the variance. Question 1. The leaf length means for set 1, 2 and 3 are 10.19 cm, 9.07 cm and 9.41 cm respectively. Compared to set 2 and 3, the mean of leaf length in set 1 is significantly larger. With a mean leaf length of 9.07 cm, set 2 has the smallest average leaf length among the other sets. At 9.41 cm, set 3's mean leaf length is more in line with set 2 than set 1. These variations in the sets could be attributed to several plant species or types, each of which has a naturally varying leaf size. Variations in leaf length may result from differences in environment conditions like nutrient level, sunlight, soil quality, and water availability. In addition, for set 1, 2, and 3, the mean of leaf width averages are 3.84 cm, 3.61 cm, and 3.68 cm respectively. Leaf width means range from 3.84 cm to 3.68 cm, and they are respectively close to each other for all sets. This shows that leaf width is more uniform than leaf length across various sets.
For question 2, what is meant by variance? Variance is calculated by taking the average of the squared differences from the mean. For example, mean width is the average width of all measured leaf, and mean length is the average length of all measured leaf. And then for the statement, the leaves have different measurements of width and length, suggests that the measurements of both width and length is vary among the leaves. This variability can be quantified using variance. Width variance is how much each leaf width measurement deviates from the average width of all leaf. And then for length variance is how much each leaf length measurement deviates from the average length of all leaf. For standard deviation is the square root of variance. For example, standard deviation of width indicates the amount by each leaf's width measurement differs from the average width, and standard deviation of length indicates the amount by which each leaf length measurement differs from the average length.
For question number 3, the reason there needs to be different sets of measurement regarding leaf width and length is that these two dimensions can vary independently and thus hold separate information about the shape and characteristics of the leaf.
For the results, the total species we have accounted is 12, but we make it as 2 quadrat, quadrat 1 and quadrat 2. So for the first quadrat, we have Bridelia species with the density 0.04 meter square. Then we have Tabernae montana corymbosa with the total number of individuals is 2 with the density 0.12 meter square. Then we have Shorea parvifolia, Molinaria leprosula, and Shorea leprosula with the density 0.04 meter square each. And lastly, we have Syzygium polyanthum with the total number of individuals is 3, and the density would be 0.28 meter square.
So here is our data presentation for quadrat 2. The site of our quadrat 2 is located at Taman Eco Rimba Ulu Bendul Negeri Sembilan. So here is our table of observation that we have made during the trip. This is the list that we have found in our quadrat 2. The first species that we have found is Garcinia xanthochymus, or locally known as Pokok Kandis. We only saw one type of this species in our quadrat 2. The density of this species is 0.04 meters squared. Next, Alcyodaphne insignis, or commonly known as Madam. For this species, there are 3 of them in our area. The density of this species is 0.12 meters squared. Moreover, we also observe Liquala spinosa, or famous known as Acarpalas, in our area. Its density is 0.04 meters squared. Then, we have also found one species of Melastoma malabatricum, or known as Pokok Senduduk Hutan, in our quadrat area. Its density is 0.04 meters square. After that, we have pokok rambai daun, or scientifically known as Shorea acuminata. And last, we observe one species of Castanopsis arganti, or locally known as pokok barangan. Its density is 0.04 meters square.
Okay, we'll continue with the question. Define the term quadrat in practice. An ecological research tool called a quadrat is a square or rectangular plot of a predetermined size that is used to isolate a standard unit of area in order to study the distribution of items over a larger area. The typical size of a quadrat is between 0.25 and 1 square meters, though this can vary based on the study's objectives and the size of the organisms being studied. Within the study area, researchers position that the quadrat frame on the ground at either randomly chosen or methodically chosen locations. The quantity and kind of organism, such as plant species, as well as other elements like the makeup of the soil or the presence of particular insects, are counted or measured within each quadrat. And then, for question 2, the two basic requirements of all quadrat techniques are quadrant must be placed at a random or systematic. Next, would be the quadrant must have a consistent and some shape and size.
Question 3, what would be the advantages and disadvantages of using a larger or smaller quadrant to survey the sedentary organism? The advantages of using a larger quadrant is more comprehensive data can be collected, reduced age effect, and better representation of sparse populations. Meanwhile, the disadvantages of using a smaller quadrant is the efficiency, detail-oriented, and can be used for focus study. Next, for question 4, the first issue that ecologists had concern regarding the sampling method conduction is spatial heterogeneity. Next is the organism's detectability. Other than that, the ecologists are really concerned about the suitability of their sampling method. And lastly, the variability of the environment.
For question number 5, according to our results, some species are present in Site 1 but not in Site 2. There are several factors that may have influenced this type of distribution. First of all, it might be due to the quality and the composition of the soil. Soil nutrients, pH, soil contents, and texture vary across quadrant. It is not the same for every area of the quadrant. Next, it may have occurred as a result of interactions between species and competition. There are also other mutualistic relationships that can affect plant presence. Lastly, it is due to seed dispersal mechanisms. There can be difference in the species composition of quadrats depending on distance from seed sources and the efficiency of dispersal agents such as wind, animals, and water.